Okay, so I'm going to start. Welcome everyone to number 6 in our IMG webinar series. Sorry, number 7 in our IMG webinar series. My name is Rekha Seshadri. I'm a computational scientist here at the JGI. And with me today, helping field chat questions are a number of scientists and software developers, but particularly Simon Roux, and I'll let Simon introduce himself.
Hi everyone, I am Simon Roux, a scientist in metagenomics at JGI.
Okay, and so hopefully everybody read the instructions and you know that you will be muted for the duration of the webinar, but if you have questions, please put those in the chat. So if you hover over the bottom as you're seeing now, you can go to chat. Oh, okay, I don't have the button, but hopefully you guys have discovered the chat button by now. And I also wanted to let everybody know that we are recording all these webinars and we will post some annotated recordings of the webinar online, hopefully before the last one next Tuesday. Okay, so I think that's plenty of time in terms of getting people on board. So the sort of requisite who are we slide. We are the Joint Genome Institute. We are funded by the US Department of Energy Office of Science. And the Office of Science funds user facilities like the JGI to enable access to large-scale science, in our case, genomics, proteomics, metabolomics, DNA synthesis. There's an expanded portfolio of products beyond just sequencing that you may or may not have heard of. We opened in 1998. We have approximately 280 members on staff, and we're located on the Lawrence Berkeley National Lab campus in Berkeley, California. And if you're interested in learning more about how to gain access to JGI's capabilities, genomics, large-scale genomics capabilities, as well as its other, portfolio of products, please visit our website, and I've provided a link below. Okay, so for today's demo, I am going to work within IMG MER. I know if you've joined webinars before, you've seen this many, many times, but the possibility that there are people joining us for the first time, we have multiple different systems, but for today's demo in particular, you will need to have a registered account with us, and you'll know that you're logged in when you see sort of this, your name appear with the logout button, and you'll be within IMG MER. And the distinction is that IMG MER provides other capabilities that we've covered, we've talked about before, like the ability to submit your data or submit larger computational requests. But most importantly, the stats tool that we're going to look at today is only available through the workspace functionality within the IMG MER system. So you will need to have access to that. I've also sent you a link to a direct link to that site in the email invitation that went out. And you can always navigate to it from workspace genome sets. Okay, so what can you do with this tool? It allows you to quantitatively compare functions across distinct groups of genomes or metagenomes that you might have curated. And you might have curated them as case versus control, where the case has a particular phenotype or a genotype, and then you have a control group. Or you might have done a sampling expedition, and you have either time series or sampling depths or some type of discrete groups. Once you have those groups curated, you can then perform a statistical analysis of differences in gene counts assigned to functions. So some real life examples from some recent publications are this one here, for example, from 2017, Vigneron et al., that compares metagenomic samples that arise from the Gulf of Mexico from distinct sampling locations. So they have site 1, which is an oil seep environment. Site 2 was a methane seep environment. And then they had their control environment, which is the sediment from outside the seep zone. Sorry, babbling today. And they performed a statistical analysis based on genes assigned to KEGG orthology terms. Okay, so this is a ternary plot. And what it shows you is individual KO terms. If they're in the middle, they're sort of, you would say, universally distributed across their samples, right? They're not enriched in one sample site, whereas terms that appear closer to one of the vertices are enriched in that sample site. And these KO terms are then colored based on the pathway that they are assigned to, right? So for example, for the methane seep, you'll see that these colors are maroon, which corresponds to methane cycle. So there's an enrichment of function for methane cycle in the methane seep, as one would expect. And similarly, there is a enrichment of functions for the anaerobic degradation of aliphatic or aromatic hydrocarbons that you would expect in an oil seep environment. So that's an example using metagenomic samples. I should point out that they probably had only about 2 samples per site, and they used a non-parametric ANOVA-based method to do the statistical analysis. These examples that showed up here just now are based on isolate comparisons. So in this case, 2 groups of isolate genomes were curated. There were thousands of isolate genomes that had been isolated from plant, rhizosphere, phylosphere, etc. So plant-associated isolate genomes. And then there was a control set of non-plant isolated genomes. And they, Levy et al., this is a really good example because they used 5 independent statistical tests to sort of arrive at their inferences. And so what you're seeing here really is a comparison based on PFAMs, where they looked at differentially abundant genes assigned to PFAMs. And one of their major inferences was that a lot of functions related to the uptake of sugars and carbohydrate metabolism in general seem to be enriched in the plant-associated group. The reason being that rhizosphere organisms need to take up sugars from root exudates from their plant hosts, for example. And this example is another isolate-based comparison comparing hundreds of isolate genomes that originate from the rumen environment, which is a sort of specialized fermentation chamber for the breakdown of plant material in ruminant animals, and comparing that to the human GI tract. And again, there are observations made that fit the expectation, such as the enrichment of carbohydrate active enzymes that break down cellulose or pectin or xylan or plant materials, and some other observations. So these are some examples I wanted to share. I think I have the citations for you in the PowerPoint presentation. So feel free to look at the method sections of each of those papers to see sort of the details.
Rekha, just a quick technical question. Is there a way to change your mouse into a pointer?
Probably.
Yeah, laser pointer probably. Let's see. Yeah, perfect. Thank you.
Thank you. Should we pause for questions? Is there any question that needs to be answered at this stage?
Nothing. Nothing that I see that would be like broad enough. So no, go ahead.
Okay. So soldiering forward. So IMG provides access to a number of different statistical tests. And what I'm displaying here is a decision tree for how the default statistical method is applied. So, if you're unfamiliar with these tests, the system will assign a default statistical method based on the data sets that you input for the statistical analysis, right? And that's informed by the number of groups. So again, IMG will allow you to curate up to 10 different or distinct groups. And based on the number of those groups, you will go up, down, one of these decision points. So if you have two groups, such as the example of the plant associated versus non-plant associated, um, you would go down this side, and then you have access to one of three different methodologies. But the default method that the group will choose, if you have more than 5 samples or more than 5 isolate genomes, if you're looking at isolate comparisons, it'll default to the Mann-Whitney test. But you always have the option of selecting one of the other tests. And really, we recommend that you certainly explore various statistical tests and try to compile a data set that can be derived from all the various different types of tests. For the methane seep, oil seep example that I just talked about in the previous slide, the option, since it had 3 groups or more, you would go to an ANOVA if you have less than 10 samples. If you have more than 10 samples, you would have Kruskal-Wallis. And then finally, when the test is performed, the UI will display the following metrics. It'll display the mean of the gene counts for each group and the standard error of each of those means, a test statistic, and then it will order the information based on the lowest p-value, the false positive rate adjusted p-value. So there is a Benjamin-Hochberg error correction that happens to generate these false discovery rate adjusted. So we're going to look at the UI very soon, and we'll have a look at one of these result sets to see what all of that looks like. Okay. Now you can compare based on 3 different features. Okay. So one of them is by function, and these are basically all the different functional annotations that are available within IMG. And you might ask, why are there so many different functional annotations? And sort of our pitch on this always is that, first of all, not all of these are completely overlapping. They have sort of different strengths, and also they tend to focus on particular research areas. And so they do have, there are some subtle as well as some gross differences between the annotation products that you'll receive from doing search against each of these databases. So we have, I'm sure everyone is familiar with NCBI's clusters of orthologous groups of proteins. Okay, we also have TigerFAMs. This tends to be a personal favorite because it has this concept of equivalogs, where they have, full length models, and it's really intended for annotation. PFAM, on the other hand, has full-length models, but it also has smaller domains or repeats. There are about 16,000 PFAM terms to sort of reflect that. And then the Kyoto Encyclopedia of Genes and Genomes, which has this additional feature of having been assigned to a pathway map. So there's advantages in each of these functional database types. CAT, SuperFAM, and SMART were recently added to the IMG annotation pipeline. And so it might not always be available depending on the date the data set was added to the UI. Okay, so a little bit about that soon. The next feature that you can compare on is an overarching function category, right? So for KEG in particular, for the Kyoto Encyclopedia of Genes and Genomes, the KO terms are actually assigned to a pathway. So I'm showing you an example here of methane metabolism in particular. So you have this really large pathway map that's comprised of up to 50 individual modules. And these modules pertain to things like, cofactor biosynthesis, CoM biosynthesis. We have methanogenesis, which a lot of people are interested in. Methanogenesis, particularly from hydrogen and carbon dioxide. You have trimethylamine metabolism. So I hope everybody understands that these pathways are broken down into these smaller subsets or modules, and there are over 160 KO terms assigned to each of these modules. And so you can now perform a test at the module level. And what that means is that the gene counts assigned to individual KO terms within a module will be aggregated before the test is performed. Okay, hope that makes sense. The third feature that you can do a test on is taxonomy, and this pertains only to metagenomic data sets. So if you've attended previous webinars or you've done any work with metagenomes or scaffolds or metagenome scaffold bins, within the UI, you might have seen a table like this. What it's shown here is individual scaffolds from, in this case, the same sample, and they're ordered by their sequence length. Each scaffold receives a sort of taxonomic affiliation. So the very first scaffold here is assigned down to the genus and species level, whereas the second one is only assigned to the phylum Bacteroidetes, and so on. And the way these scaffold lineage names are assigned is based on this majority hits approach, right? So in this example, I have a scaffold that has only 6 coding sequences on it. And if you look at the last hits, 2 of those have no hits, and 4 of them have hits, but they are to different genera within the same family. And the sort of rule of thumb is that we need to have a majority. So over 50% of those coding sequences need to be assigned, to one of these genera to receive that assignment. So in this case, since that can't be resolved, the scaffold then receives the name family Acidobacteriaceae. Okay. Okay, I'm going to pause there. Any questions about that, Simon?
We are going through the question, but I don't see anything. No, I don't think anything that would be directly relevant to what you're doing. So I guess moving on and getting to the live demo will answer most of this question, actually.
Okay, all right, let's get there. Okay, so I have a quick how-to guide. You have access to all these slides. For the demo purposes today, I'm going to look at a 2-group comparison, and it's going to be based on metagenomic samples, since a lot of people seem to be interested in metagenomic analysis in particular. So they kind of won out by voting for metagenome-based webinars. So I'm going to be comparing samples from the same study. We have 30 samples that originate from Arabidopsis rhizosphere. And within that same study, they had a control set of about 14 samples that came from unplanted soil. And so we're going to be doing a statistical test on these 2 groups. So as I've already mentioned, you can actually curate up to 10 different sets of either metagenomic samples or isolate genomes. Once you carefully curate, and there's a reason why carefully curate is bolded in a minute. Once you carefully curate these sets, you can add those sets in your workspace under genome sets. And keep in mind that you can include, especially for isolate group comparisons, thousands of isolate genomes, and even hundreds of metagenomic samples. But of course, the larger your job is, the more computationally intensive it's going to be, and the longer it's going to take for the results to be presented. So the carefully curate, really, and I'd like to nail this point home. In computer science, people like to use this term garbage in is garbage out. And so really, you really have to make sure that the data sets that you're curating are not flawed in any way, right? You want to make sure that you have relatively clean data that's not noisy, and it's not going to introduce any artifacts, because at the end of the day, you might get statistically significant results, but that doesn't mean that you can make any sound inferences based on any flawed, if there's flawed input, there's going to be flawed output. So just be careful about that aspect. Once you've created these genome sets or metagenome sets, you can then go to the statistical analysis tool. We have a user guide. And then once the test is launched, you just have to wait for an email to notify you that your job is completed. If you're impatient and want to see what's going on, you can also go to Workspace and check in My Jobs for the status of your job. Okay, so at this point, I'm going to move over to the UI. And we're going to okay, Simon, can you see the UI?
Yes, we can.
Okay, great. All right, so I am in IMG MER. I am logged in. And I'm going to navigate, actually, I'm not going to navigate there first. We're just going to very quickly review the advanced genome search again. The very first webinar in the series was the advanced genome search. And if you go to the webinar registration site, you'll actually see the recording of the video. The recording of the video is also available here if you want to learn about all the features and all the different types of metadata that you have to search across. But for this example, I'm just going to very quickly go to pull up these data sets. And really the purpose here is to show you how to create a genome set in Workspace, right? So I'm going to pull back microbiomes, and I want them all from a particular study that I've checked out before. And so I'm just going to say Arabidopsis here. I'm going to add an additional filter so that I only bring back metagenomes and not metatranscriptomes, because there are metatranscriptome samples that'll get retrieved otherwise. If you're interested in this particular metagenome analysis, all right, and I'm not going to evaluate, I'm just going to search. But if you're interested in this particular query that I'm using here and you want to sort of follow along with me, all you have to do is go down and click on this View Public List. Again, I think I put the information in the slide that I sent out, and you can load this query and you can hit search right along with me. Okay, so I'm retrieving metagenome samples. It looks like I get 161 that meet all my criteria of it being a metagenome and having study name Arabidopsis, right? And so hopefully you've attended some of the webinars before, and you've sort of familiar with this concept of what these tables look like, the fact that they can be reconfigured in different names. So I'm going to hide, say, some of these columns that I'm not interested in. These were all sequenced here at the JGI. I'm going to remove some of these IDs. So I think it was webinar number 2, where Natalia goes over even how to filter these using regular expression searches. Okay, and then all the good stuff is at the bottom of the table, as we always like to say. So you can reconfigure that table above with all these different metadata types. If you're interested in statistics, if I've sort of auto-checked if there are any bins, but you might be interested in other pieces of information. Maybe you want to look at all the metadata fields. And hopefully all of this will just assist in the curation efforts. And you just hit redisplay and you get a table. So I'm kind of sort of pre-configured this table, so I'm sort of satisfied with what I have here. And as I said, my objective here really is to show you how to create a set in Workspace. So for that, I'm just going to reiterate, so you can see that there's some of these metadata types that's telling you that these samples come from rhizosplane, from Arabidopsis thaliana, and I'm interested in samples that, for the control, which come from unplanted soil. I'm just going to filter this with unplanted and hit apply. And I'm going to select all those samples. So that's my 14 samples. That'll be my control set. And at this stage, you're probably familiar with the add selected a genome cart button, but right above that is save to workspace. Okay, so I'm going to hit save to workspace here. It just takes me to the bottom of the page, and I'm going to say, these samples were from North Carolina, and this is the unplanted soil set. I give it a name, and I just say save selected to workspace. And so here, this breadcrumb tells you where you are. It's taken you to workspace, and it's put you in the genome sets part. And we are going to now do a statistical analysis. So very carefully, I've created one of the sets. Obviously, I've created my case or my Arabidopsis rhizoplane set before. And as I said, that's the part that's actually the one that requires the most amount of attention, because you want to assess you want to assess the quality of those data sets. You want to assess the metadata of those data sets to make sure that it all looks like it's cohesive. And so I'm now going to navigate over to statistical analysis. Are there any questions, Simon, about what I just did in the advanced genome search?
I mean, I guess one is, can you remove a genome from a genome set once you put it in?
Oh, the answer is yes. It's a little more, I don't know if I should go over that. Okay, let's go over that real quick. It's a little more convoluted, I guess. You would have to go to your, I'm just going to pick this one, for example. You would have to go to your set. So I'm pulling back, I think, 46 isolates in this case. And let's say I don't want these two guys. I would then, okay, I guess it's not that simple. You would have to go back to the genome cart and then add a new, this is, perhaps Natalia might have a better answer for you, but I think in the interest of time, I'm going to go back to what I know, what I feel comfortable with in terms of navigating in the UI. But I think you might have to recreate
from the genome cart.
Yeah, from the genome cart. But you can actually just, I think, replace an existing workspace set, if that makes sense.
Okay, sounds good. Thank you.
Okay, so here we are in the statistical analysis tab. And as I mentioned before, we have this user guide, and you can peruse these if you've you want a quick guide. This is the decision tree that I mentioned before. There's some additional information here about each of the tests and what their sort of limitations or what their underlying assumptions are. And there's some additional details here for people that are really interested in the minutia. Okay, so my two sets are right here. I have my North Carolina unplanted soil that I just created, and this I've created before, which is Arabidopsis rhizosphere, and I have 30 samples and I have 14 samples. The next is selecting the features that I just mentioned. So you can select by function, right? And you have all these different functional assignments, or by function category, or by taxonomy, which is relevant here because I have metagenomic samples. Now this question mark is important. What this lets you know is when these data sets, or when all of IMG data sets might have been, what version of databases they might have been annotated. So as I mentioned before, Superfam, SMART, and CAT, for example, were added only in March of 2019. So these would not be available for data sets before that. So for the examples that I have here today, these were generated in 2014. And I know for a fact that these four categories will not, are not annotated, and so we can't use that. So this is here as a guide to sort of give you an idea of when the annotations might have happened, or that's again something that you would have to assess yourself for the data sets that you have, but it's telling you as you saw for each data set, when the annotation might have happened and what database um, of the annotation source was used. Okay, the next is the measurement. So here again, this is pertinent for metagenomes. When you're looking at just isolate genomes, you would go with just gene counts, but because we have metagenomes, and I've ascertained that for my particular metagenomes, indeed we do have read depth information, so we can use this estimated gene copies feature, right? So in this case, very simply, we have a glossary of terms that explains this as well. Basically, you take the genes and you multiply that with the average read depth of the scaffold that they reside on. Okay, so because we have metagenomic samples, I'm using estimated gene copies. I should also point out that if you're comparing data sets where some data sets have estimated copies and other data sets do not have estimated copies, then the UI will tell you that, will throw an error message saying that this is not possible because you've selected estimated gene copies and some of the samples in your groups do not have that information associated with them. Okay, that's sort of our caveat about this. The statistical method, we've already covered. The system will default to the default method, and then you have the opportunity to set a different method as well. The relative and absolute, again, is something that's relevant to if you're doing an isolate-based comparison or metagenome-based comparison, right? So if you have if you have isolate data sets and you're interested in looking at presence and absence across groups, right? You're just looking at frequency, a preponderance of presence in group 1 versus a preponderance of absence in group 2, you would select absolute. But that's not what we're doing here. We're looking at metagenomes, so we're interested in a relative comparison where the gene counts are normalized for the total size of the data set. Step 4 is choosing the display options. Okay, so you can choose to show all rows. So in this example, let's say I use KO, right? I think there's over, I think close to 13,000 KEGG orthology terms. So I can show the results of all 13,000 KO terms, or I could say, I only want to see rows with at least one non-zero count. So this means that, non-pertinent KO terms that come from maybe the eukaryotic side, we don't really want to see those. We don't want to see rows with all zeros, so that's what you do here. The last is a little more prohibitive, where it'll only show results with what we've deemed as, and we've set significant hits at a p-value, adjusted p-value of less than 0.05. But I would default to maybe this one, or maybe even this one. And just put a job name, North Carolina, Arabidopsis rhizosphere versus unplanted soil, right? And we hit run analysis, and it's telling me that the default method, Mann-Whitney method, will be used for my particular comparison. And as I said, you will get an email letting you know when your job is completed. For a data set of this size, I've had the results returned to me in just a couple of hours. Okay, so I'm going to go and look at my completed results. So I just go to Workspace, My Jobs, and you'll get a list of all the different computation jobs that you've submitted. I'm just sorting this table by the most recent jobs, and so you'll see here that these are all the tests that I've submitted relevant to these particular groups. So I've done based on KO terms. I've done based on taxonomy class. I've used KEGG modules. I've used PFAMs, and I've used family based on taxonomy again. So let's take a look at one of these real quick. I'm going to look at the test that I performed on rhizoplane, 30 samples versus 14 unplanted soil samples. And okay, very important to highlight here. Only the first 1,000 rows are shown in the UI. Okay, So this is not a problem in this particular case because there are less than 1,000 rows, because we're looking at class. But if you had something like KO terms that has, or PFAMs that has 16,000 plus terms, uh, beware that only the first 1,000 hits will be shown. Okay. If you want to see the complete set of results, you would go to download full results. And I'll display that in just a minute in the next example that I'm going to go over. But for this particular example, the table has class as your feature name. The description of the class is the complete hierarchy um, for that particular class. And then you have the mean, excuse me, you have the mean of the counts arising for that particular class from 30 samples, and then the mean for the 14 control samples, the standard error for each of those means, the U-stat, the p-value, and then the adjusted p-value. And so as I said before, this table is ordered by the most significant hits. You can sort this the other way and you can sort of get a taste for, well, clearly there are things that are not significantly differential at all between the two groups. And you can filter this as before using text or regular expression searches. So, my expectation, since we're comparing rhizosphere or rhizoplane samples against control bulk soil samples, is that I would expect to see lineages that I know are plant associated, like rhizobacteria and such. So I'm just going to very simply filter my class with a partial search. I'm just going to say rhizo and hit apply. And so that's pulled back class rhizobiaceae and Brady rhizobiaceae. And sort of confirming my expectation, I see that there seems to be the means are certainly larger in my Arabidopsis rhizosphere versus the unplanted soil. And my adjusted p-values are also very significant. So that's something that's sort of confirming the hypothesis. Okay, so I'm going to go to another example, and I want to show you the results of the download full results in particular. So I'm going to go back to my jobs and workspace. And I'm going to find the results of the test from KEGG orthology terms, right? So I'm going to click on that. And so we have the same table with all the same information, but now the feature is my KEGG orthology term, and then the name assigned to that term, and then again, all the same metrics. And going back to this issue of only 1,000 rows are shown, right? So the top 1,000 are shown. It's possible that I have more than 1,000 hits. That might be. So this is the highest p-value in my list of 1,000 is 0.005. So if you go with the the usual rule of thumb, which is 0.05, that's a much bigger discussion than we'd like to get into here. But if you want to see more hits beyond the 1,000, you want to go to download full results. And what this will provide is a compressed file. And so let's just go take a look at that real quick, okay? I'm just going to uncompress that file. And I'm going to open it. I'm going to pull out UI data output and just opening it with Excel. And there it is. Okay, so what you have in this table now is beyond the 1,000, we have, I think I select all non-zeros. Yeah, so I have 8,410 KO terms now displayed. And then here is the adjusted p-value, right? here. So you can see the ascending and the descending. You see the non-significant hits are here. And then the other thing I want to point out to you is that you have the normalized counts of every single sample in every group. So for my 30 plus 14, 44 different samples, I have the normalized counts of the genes. And then I have followed by those 44, I have the raw gene counts as well in this table. So this is a really good resource, especially if you want to um, check out other statistical methodologies or other ways of normalizing. This is also a resource that's available. So let's go back to our table. And so this is the fun part, right, where you get to sort of hypothesize. This is very interesting to me. So I'm familiar with this aminocyclopropane carboxylate. This is ACCase. It's a pretty well-known plant growth promoting determinant, and certainly the mean counts seem to suggest that it's overrepresented in the rhizosplane samples as opposed to the unplanted soil. And if you peruse this list, of course, there's going to be a lot of sugar transporters and sort of similar to the observations that have been made before in the literature. The last thing that I want to point out with this is you can also select, you can come up with your own cutoffs and maybe sub-select. But in this particular case, I have 1,000 hits of roughly 0.005 and lower. So I'm just going to select all of them. And I am going to add these 1,000 functions to my function cart. And the reason I do that is, it's impossible to know where all of these functions fit in terms of metabolism. And you tend to sort of zero in on the things that you're familiar with. But the advantage of this is that since we're working with KO terms, we can actually see what particular KEGG pathways those particular enriched KO terms are assigned to, right? So that, it's just you can see a lot of ABC transporters, and a lot of those are the sugar transporters that that I've mentioned before that came out of that isolate-based comparison as well. So another one of the things that people like to look at, particularly from sort of a plant growth promotion or plant beneficial process, is nitrogen metabolism, nitrogen fixation in particular. So that then shows you where some of those KO terms are assigned, and just sort of, in the context of a pathway map. So I just wanted to, so if you have nitrogen metabolism down here, for example. Okay, so that was I'm at 11:44. That's where I wanted to sort of end. So Simon, do we have questions that are?
Sure. There is one that is both very interesting and also kind of a trick question, so I apologize in advance. But
Well, you're here to help me answer them.
I mean, it's because we don't have a good answer for it, but it's an interesting one.
Okay.
Is it possible to restrict features to a specific taxon? For example, can we compare KOs between groups of metagenomes using only scaffolds that have been annotated as archaea?
Ah, what a beautiful segue into
Right?
What a nice segue into and a nice reminder for me to go back to my slides. Okay, so the answer is it's coming soon. So if we go here, okay, so it is currently, it has been developed already. We've especially now that we have these auto-generated scaffold bins, the next logical thing is, okay, and also I want to mention that we've been working very hard on a bulk download of those, so that is also in the works. But we do have the ability now to use scaffold bins Or in this particular example, it would perhaps not be the automated scaffold bins, but a scaffold set that you would curate yourself, where it's all assigned to a particular taxonomy, and you want to do a comparison based on that. And the answer is yes. It is in beta testing, and it'll give you the ability to do, in this particular use case example that Simon just stated, as well as the ability to use the metagenome scaffold bins and sort of these genome equivalents and have these mixed groups where you have both the bins with isolate genomes, very carefully curated. So, as I mentioned, that's coming very soon. Beyond that, we're also developing an additional statistical tool, especially for isolates or genome equivalent comparisons, where we'll be using um, um, basically phylogeny informed generalized linear models. So thanks for the setup, Simon. Do you have anything else to add?
I mean, it's another question that is probably worth addressing live, although it's slightly tangential to this statistical tool. But since the statistical tool is all based on annotation, how efficient is this tool for genome description and annotation of novel species? Which I guess is another way of saying, if I have very, very new species, what should I be careful with when using, let's say, comparison of KO or PFAM?
Yeah, I mean, the short answer is you're out of luck. But that's not okay. So, I mean, yes, I mean, the UI is not a one size fits all solution. I guess in that particular case, if I'm being honest, I'm doing it myself, I would probably want to, generate gene clusters myself and then do a statistical test. Maybe I have lots of conserved hypothetical proteins that do not match any of these functional annotation databases. But that being said, I mean, the existing, repertoire of functional annotations that we have do, especially, and I can talk to isolates in particular, they do have good coverage, right? So you can have almost 80% of all coding sequences in your isolate genome that are assigned to these categories. But if you're asking about, the new, right? The 20% of hypothetical or conserved hypothetical proteins, you'd have to do it outside of the UI at this stage.
Mm-hmm. And that's pretty much it, yeah.
Okay. Well, thank you everybody for joining us. Please use the contact us button in the UI if you have further questions. We will be posting the the Q&A session today. If we didn't get around, if there are too many questions and we didn't get around to answering everything, we'll probably work on it and provide a more thorough answer, and we'll post those on the MGM webinar site. And also, please check out our community sequencing program, particularly the new investigator call. We have a due date in September, so just explore to see if you can sort of gain access to JTI's capabilities. And that's all, folks. Thank you.